Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to the Honorable Dean of the Faculty of Science and Technology, Universitas Aisyah Yogyakarta, Mrs. Hapsari Wahyuningsi, Sarjana Teknik, Master of Science. Deputy Dean of the Faculty of Science and Technology, Mrs. Nosa Septiana Anindita, Sarjana Peternakan, Master of Biotechnology. The head of Biotechnology Study Program, Mr. Arif Bimantara, Sarjana Peternakan, Sarjana Perikanan, Master of Biotechnology. Our distinguished guest, the Dean of Faculty of Industrial Sciences and Technology, University Malaysia Pahang, Dr. Saiful Kamaluddin Muzakir Elias Lukman, Deputy Dean of Academic and Student Affairs, Dr. Muhammad Fazli bin Farida Asras, Deputy Dean of Research and Postgraduate Studies, Professional Technologist Dr. Azizan bin Ramli, the Head of Industrial Biotechnology Program, Dr. Lee Chin-mei, and to all our invited speakers, lecturers, students, and dear participants, on behalf of the organizers, the Faculty of Science and Technology, Universitas Aisyah Yogyakarta, and the Faculty of Industrial Sciences and Technology, University Malaysia Pahang, we would like to wish you a warm welcome. Selamat datang to this exciting program, the Virtual International Summer School, Science and Culture UNISA-UMP. I'm Marwah Amalia, a biotechnology student from Universitas Aisyah Yogyakarta, will be your MC for today. This week program aims to provide students with interesting topics on applicative biotechnology, fun little experiments showcases, as well as virtually exploring local places from each country. This program would also increase students' engagement and interest towards bio-entrepreneurship. We feel honored and privileged to serve the best to you through this exciting program. I would like to thank you all for participating in this opening ceremony. First of all, let us thank Allah Subhanahu wa Ta'ala who gave us blessings and health during this time so that we were able to attend this opening ceremony. Let us also send our prayers to our Prophet Muhammad Sallallahu Alaihi Wasallam and his progeny. To begin the opening ceremony with the blessings, I would like to invite Mr. Muhammad Arif Pikahulan to lead the dua recitation and a moment of silence. To Mr. Arif, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. Ya'lamu ma baina aydihim wa ma khalfahum wa la yuhittuna bihi ilmi. وعنات الوجوه الحي الكيوم وقد خاب من حمل ظلما ومن يعمل من الصالحات وهو مؤمن فلا يخاف ظلما ولا هضما وكذلك أنزلناه قرآنا عربيا وصرفنا فيه من الوعيد لعلهم يتقون أو يحدث لهم ذكرا فتعالى الله الملك الحق ولا تعجل بالقرآن من قبل أن يقضى إليك الوحي وقل ربي زدني علما ولقد عهدنا إلى آدم من قبل فنسي ولم نجد له عزما صدق الله العظيم.
Thank you for the dua recital, Mr. Arif. Dear participants, ladies and gentlemen, I would like to invite all of you to join us singing the March Song of Universitas Aisyah Yogyakarta and to be followed by the March Song of University Malaysia Pahang.
Wahai seluruh mata UNISA Yogyakarta, bulatkan semangat baktimu. Siapkan generasi unggul dalam prestasi, berjiwa profesional berani. Iman ilmu kuatkan, amal mulia kerjakan, akhlakul karimah wujudkan. Sumsum fajar pencerahan timah, sejahtera bumi bersada. Kembangkan sumber daya manusia, berkemajuan capai cita. Itulah misi kita bersama sebagai
Thank you. Now let's begin with a welcome speech. Please be invited the Dean of the Faculty of Science and Technology, Universitas Aisyah Yogyakarta, Mrs. Hapsari Wahyuningsih. The time is yours.
Ya, terima kasih Mbak Marwah Amalia. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Assalatu wassalamu asrofil anbiya'i wal mursalin wa ala alihi wa ashabihi ajma'in amma ba'du. The Honorable Dr. Saiful Kamaluddin Muzakir, Dean of Faculty of Industrial Sciences and Technology, University Malaysia Pahang, all Deputy Dean UMP, Pak Arif Bimantara as Head of Biotechnology Study Program, all speakers, all lecturers from University Malaysia Pahang and Universitas Aisyah Yogyakarta, and also all participants, all students from UMP and UNISA. Good morning, everyone. It is a great pleasure for me to welcome you to Campus UNISA Yogyakarta virtually. On behalf of the Faculty of Science and Technology, I would like to welcome all of you to this morning event, International Summer School Science and Culture, UNISA and UMP, that will be held into 4 days. This International Summer School with topic Science and Culture, UNISA and UMP 2022, it's about practicing of biotechnology in daily life that respect the local wisdom of each country, Malaysia, also Indonesia, of course. One of local wisdom that we will learn today, or maybe in the 3rd day of this International Summer School, is about tempe. Tempe as a traditional food in Indonesia, and we know that Indonesia is the largest tempe producer in the world and the largest soybean market in Asia. More about tempe and the history of tempe and about the culture will be delivered in the 3rd day. And in the last day, we also will going around tourism village at Yogyakarta virtually, of course. And the tourism village that we will visit it virtually is one of the tourism village in Yogyakarta that produces salak fruit, buah salak, ya. The biotechnology study program has facilitated the development of the tourism village by using grant funds from the government last year, especially for various kinds of food processing from salak fruit. So hopefully in the next agenda can be held directly on site, offline, so all of us can feeling the experience at the tourism village directly. This international summer school attended with by approximately 80 participants, yeah, from both UNISA and UMP. So I want to say many thanks to all committee, seluruh panitia who have helped organize this event very well, and I hope the event runs smoothly until 4 days until the end. I hope this collaboration between Faculty of Science and Technology UNISA Yogyakarta with Faculty of Industrial Sciences and Technology University Malaysia Pahang will be sustained and bring a lot of benefit for all of us. Saya rasa sebelum saya menutup, izinkan saya berpantun. Duduk meja sambil makan, pemandangannya hutan ilalang. Beribu terima kasih kami haturkan teruntuk saudara-saudari yang telah datang. Sungguhlah tampan putra mahkota, sungguh bijaksana tidak lalai. Dengan basmalah pembuka kata, pertanda acara akan dimulai. Cukup sekian, saya kembalikan ke Mbak Marwah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Hapsari, for the remarkable welcome speech. To follow, the next welcome speech by the Deputy Dean of Research and Postgraduate Studies, University Malaysia Pahang. Please be invited, Mr. Dr. Azizan bin Ramli. You may present your speech followed by opening the International Summer School program.
Thank you very much, Ms. Chairwoman. So, Assalamualaikum warahmatullahi ta'ala wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
اللهم صل على سيدنا محمد الفاتح لما أغلق والخاتم لما سبق والناصر الحق بالحق والحادي إلى صراطك المستقيم وعلى آله وصحبه حق قدره ومعداره العظيم. So I will now like to make a speech on behalf of our Dean, Associate Professor Dr. T.S. Saiful Kamaluddin. So since he have another agenda today, so he only expressed his warmest greeting to all of you, and very sorry for not being able to attend this, this very memorable event. So thank you very much to the organizer, because they already prepared the text for me. Okay, so I'm just reading the text on behalf of our Dean. The Honorable Deans of Faculty of Science and Technology, Universitas Jakarta, or in short, UNISA, Indonesia. The Deputy Deans, Faculty of Science and Technology, Universitas Jakarta, Mrs. Nosa Septiana Anindita. And then the Head of Biotechnology Study Program, Mrs. Arif Bibantara. I'm very sorry for not mentioning, I forgot to mention the name of the Honorable Dean of your faculty, Mrs. Hapsari Wahyuningsih. I'm very sorry for that. So ladies and gentlemen and the fellow students, salam sejahtera and very good morning to all of you. I'm honored to be here today. I would like to welcome all of you to this virtual summer school, Science and Culture, UNISA-UMP. Jointly organized with the Department of Science and Technology, Universitas Aisyah Yogyakarta, Indonesia. The International Summer School with the term Science and Culture, UNISA-UMP, is an initiative platform for bridging FIS, UMP, and the Department of Science and Technology in academics that involve undergraduate students. Fellow students, we are very fortunate to have this event, which has been organized for 4 consecutive days, starting today until Tuesday. The participants involved in this summer school are undergraduate students from both UNISA and UMP. And the main objective of this event are to develop a culture of economic collaboration in multicultural environments that involve international speakers and students, specifically from UNISA Indonesia and UMP Malaysia, to increase the knowledge and improve the academic and scientific understanding of students from the sharing of knowledge by speakers from both UNISA and UMP. We hope this program will motivate all of you and increase your interest to explore scientific knowledge of biotechnology that contribute to bio-entrepreneurship ideas in the future. And last but not least, we hope that all the students will have a positive attitude forward to learning and gaining knowledge during this program through activity learning, as well as learning about the culture and making new friends. Ladies and gentlemen and fellow students, FIS is also active in arranging student exchange program and student mobility, including an inbound and outbound programs from and to several countries respectively. FIS has sent several students to spend 6-month internship in industries abroad for the internship program. In the past, UNISA has actively arranged and been involved in several activities organized by UMP. For instance, student exchange and participated in the International Festival of Innovation and Green Technology, or IFINO. This year, UNISA also planning to send their students for mobility to FIS UMP under the Industrial Biotechnology Program. Following this, to maintain the momentum of good collaboration, UMP and UNISA wish to strengthen cooperation between institutions through organizing the events today. To this event, I'm delighted with the participation of more than 80 students from both UNISA and UMP. FIS always recognized the importance of international collaboration in many aspects, and we hope that this event will open more opportunities in future. Again, I hope that we will use this opportunity to continue an engagement between UMP and UNISA in exploring the new possibility of some future events. Before ending my speech, I also wish to express my sincere appreciation for the excellent cooperation from lecturers and staff of the Faculty of Science and Technology, UNISA and S.Si. This summer school will not been without this summer school will not been possible. To all students, I'm sure this event is going to be learning experience and wish you all a very fruitful deliberation throughout this International Summer School Science Culture University UMP. So thank you very much. Wabillahi taufiq wa hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Dr. Azizan bin Ramli, for the outstanding welcome speech. So, dear participants, ladies and gentlemen, the virtual International Summer School Science and Culture UNISA-UMP is now officially opened. By this, it is the end of opening ceremony. I'm thankful to all guests and participants for attending this ceremony. We wish you all a productive, fruitful class session in this 4 days program. Most importantly, do enjoy every section of the program. I, Maroh Amalia, signing off as your MC today, and I would like to invite Ms. Sarfina Mutia Sharifah, our program moderator. Welcome to you, Miss, and I will pass my duty from now to you.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Does everyone clearly hear my voice?
Yes, Miss.
Okay. So before we start our program for today, thank you all for all the distinguished guests, ladies and gentlemen. So before we start for our first session, we will play a video of Biotechnology UNISA. So by then, afterwards, I will announce regarding the rules and regulations and also the housekeeping information for all the dear students.
Assalamualaikum warahmatullahi wabarakatuh. Selamat datang di program studi Biotechnology Universitas Aisyah Yogyakarta. Alhamdulillahirrabbilalamin, pada tahun ini kami berhasil memenangkan hibah kerjasama dan implementasi kurikulum Merdeka Belajar Kampus Merdeka yang dipersembahkan oleh Probowati. Pada penerapan kebijakan MBKM tersebut, kami melaksanakan 3 bentuk kegiatan pembelajaran. Yang pertama adalah pertukaran pelajar yang bekerja sama dengan Fakultas Teknologi Pertanian Universitas Brawijaya Malang. Lalu yang kedua adalah kegiatan penelitian yang bekerja sama dengan Badan Riset dan Inovasi Nasional atau BRIN. Lalu yang ketiga adalah kegiatan magang yang bekerja sama dengan PT Biotech Cipta Kreasi. Besar harapan kami dengan diimplementasikannya kebijakan MBKM di program studi Biotechnology Universitas Aisyah Yogyakarta benar-benar akan mewujudkan link dan match antara kompetensi mahasiswa dengan kebutuhan masyarakat saat ini.
Perkenalkan, nama saya Muhammad Nasib. Saya dari Universitas Aisyah Yogyakarta. Saat ini saya mengikuti program MBKM yang dilaksanakan di Universitas Brawijaya Malang. Program pertukaran pelajar merupakan program baru yang dikeluarkan oleh pemerintah Kemendikbud. Program ini membuat kita lebih mengenal dengan kampus yang lain dan juga ilmu yang lebih luas.
Halo, perkenalkan nama saya Sabrina Kurota Ayun. Saya mahasiswi dari Universitas Aisyah Yogyakarta, program studi Bioteknologi. mengikuti program ini, saya mendapatkan lebih banyak pengalaman dalam belajar, bertemu banyak teman, kemudian mendapatkan suasana belajar yang baru, dan tentunya juga bertemu dan mendapatkan ilmu dari banyak dosen hebat kita.
Assalamualaikum warahmatullahi wabarakatuh. Saya Tunjung Mahatmanto, Ketua Program Studi Biotechnology di Universitas Brawijaya. Alhamdulillah, program MBKM pertukaran pelajar dengan program studi Bioteknologi Universitas Aisyah Yogyakarta telah dilaksanakan dengan baik di beberapa mata kuliah yang diselenggarakan oleh Fakultas Teknologi Pertanian Universitas Brawijaya pada semester ganjil 2021-2022 ini. Kami berharap kerjasama yang baik ini dapat diteruskan dan diperluas cakupannya sehingga memberi manfaat yang lebih besar bagi seluruh sivitas akademika di kedua universitas. Kepada para mahasiswa, tetap semangat belajar, memperdalam ilmu, dan memperluas jaringan melalui berbagai program MBKM yang ditawarkan. Semoga keberkahan dilimpahkan atas upaya kita membangun Indonesia melalui program MBKM yang kolaboratif ini. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Nama saya Satrio Krido Wahono. Saat ini sebagai PLT Kepala Kantor Balai Penelitian Teknologi Bahan Alam Badan Riset dan Inovasi Nasional. MBKM sendiri secara umum bertujuan untuk mensinkronkan antara dunia akademis dan dunia kerja. Sinkronisasi ini dilakukan dalam rangka untuk penerap teori-teori yang diperoleh di dalam dunia akademis secara praktek di dunia kerja. Bagi dunia kerja sendiri, manfaat dari MBKM adalah terutama bagi kami di BPTB ABRIN. Yang pertama, kami mempunyai tugas untuk menjalankan manajemen talenta inovasi nasional, dalam hal ini menyiapkan kader-kader bangsa untuk ke depan menjadi peneliti-peneliti atau menjadi inovator-inovator yang nantinya akan mengisi pembangunan di Indonesia. Yang kedua, tentu saja dengan adanya MBKM ini, mahasiswa-mahasiswa yang nantinya menjalankan MBKM dapat menjadi agen untuk transfer knowledge terkait dengan apa-apa yang kami hasilkan di BPTB ABRIN ke masyarakat.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Aulia Uswatun Hasanah dari Program Studi Bioteknologi Universitas Aisyah Yogyakarta. Saya di sini mengikuti kelompok penelitian teknologi bahan alam. Pengalaman yang dapat saya implementasikan ke depan yaitu saya mampu meningkatkan keterampilan dalam dunia kerja. Saya juga mampu mengasah keterampilan dari segi teknis maupun non-teknis. Dari segi non-teknis, saya lebih bisa mengatur waktu, disiplin, bertanggung jawab, dan beradaptasi di dalam lingkungan yang baru. Untuk segi teknis, saya mampu mengoperasikan beberapa alat yang belum pernah saya operasikan sebelumnya. Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, saya Putri Nabila Naziro dari Program Studi Bioteknologi Universitas Aisyah Yogyakarta. Dalam MBKM ini dapat membantu saya untuk mempermudah penyusunan tugas akhir saya dalam berbentuk skripsi.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, saya Amelia Sanwatita Fitri dari Prodi Biotechnology Universitas Aisyah Yogyakarta. Harapan saya terhadap program MBKM, mahasiswa bioteknologi bisa melakukan PKL dan penelitian di BRIN dengan kota yang lebih banyak.
Assalamualaikum warahmatullahi wabarakatuh. Nama saya Muhammad Ain Syarah Rahab, salah satu peneliti di Balai Penelitian Teknologi Bahan Alam Badan Riset dan Inovasi Nasional yang bertempat di Gunung Kidul, Yogyakarta. Saya berasal dari kelompok penelitian teknologi bioaditif pakan. harapan untuk program MBKM ke depan adalah ini lebih banyak lagi yang mengikuti. Nantinya mahasiswa akan memiliki keunggulan skill terhadap alat-alat di laboratorium yang lebih baik dengan fasilitas yang ada di dalam BRIN. Program MBKM ini akan memberikan jembatan atau menjembatani mahasiswa dari dunia pendidikan ke dunia bekerja. Dan di sini contohnya adalah lembaga riset, di mana nanti mahasiswa tadi sudah mengetahui kondisi lingkungannya bagaimana pola bekerjanya, dan juga sudah dibekal dengan skill-skill dari program MBKM di lembaga riset tersebut. Perkenalkan, nama saya Wisnu Adi Susila. Saat ini saya mendapatkan amanah sebagai Head R&D dari PT Biotech Cipta Kreasi. Menurut saya, implementasi program MBKM sangat bermanfaat bagi industri bioteknologi di Indonesia. Selama 6 bulan, mahasiswa akan melatih kemampuan soft skill dan juga kemampuan teknis mereka, dan juga selama 6 bulan tersebut, perusahaan bisa memantau perkembangan mereka, sehingga ketika nanti mereka lulus kuliah, mereka menjadi talent ready yang siap digunakan oleh perusahaan. Harapan saya supaya implementasi program MBKM ini terus dilanjutkan, karena akan memberikan dampak yang positif bagi perkembangan industri di Indonesia, utamanya industri bioteknologi. Dan kemudian harapan saya adalah dengan ilmu yang diperoleh di sini akan terbentuk startup baru yang akan menjadi raksasa bioteknologi di Indonesia dan juga di dunia.
Halo teman-teman, perkenalkan nama saya Radrikan Rahmatia, biasa dipanggil Ferlin. Saya adalah mahasiswi Universitas Aisyah Yogyakarta, mengambil program studi bioteknologi. Saat ini saya mengikuti program MBKM magang di PT Biotech Cipta Kreasi, masuk ke dalam divisi R&D. Pengalaman yang dapat diimplementasikan ke depannya yaitu saya mengetahui teknik-teknik isolasi bakteri dari mulai pengambilan sampel hingga ke karakterisasi isolat. Dan tentunya pengalaman tersebut sangat bermanfaat bagi dunia kerja ke depannya.
Halo, perkenalkan nama saya Rifka Sulistiawati. Saya berasal dari program studi Bioteknologi Universitas Aisyah Yogyakarta dengan divisi produksi di PT Biotech Cipta Kreasi. Pengalaman yang saya dapatkan selama magang di sini adalah saya bisa terjun langsung ke dunia kerja, serta saya dapat mengimplementasikan ilmu yang saya dapatkan selama kuliah, sehingga harapannya saya bisa meningkatkan pemahaman serta kapasitas dalam diri saya.
Perkenalkan, nama saya Maratil Kabatia dari Prodi Bioteknologi Universitas Aisyah Yogyakarta, dan saya magang MBKM di PT Biotech Cipta Kreasi dan masuk divisi produksi. Kenapa saya memilih magang program MBKM? Alasannya yaitu karena untuk memperdalam serta meningkatkan kemampuan kompetisi di dunia nyata yang sesuai dengan potensi bakat dan minat saya. Pengalaman saya setelah mengikuti program magang MBKM ini yaitu soft skill saya meningkat khususnya di bidang bioteknologi, di mana saya bisa memanfaatkan bagaimana caranya mengolah pupuk agar bisa dimanfaatkan di bidang pertanian khususnya untuk masyarakat. Harapan saya untuk program MBKM ini kedepannya bisa terus berlanjut dan bisa dirasakan oleh semua mahasiswa di seluruh Indonesia.
Okay, sorry. Okay, thank you for the time. Okay, sorry, my mic was muted before. So before we start our next session, perhaps we could take a photo first before we start our first session, because I actually forgot to ask the MC to take a photo before this one. So, okay, I hope all of the audience could open their cameras so that we could take a short picture together, especially dear students. Okay. Okay, we start from the first slide. Okay, wait. Okay, 1, 2, 3, smile. All right, the next one, the next slide. I think I still see lots of students haven't opened their camera yet. Please do so. Okay. Okay. Okay, the next slide. 1, 2, 3, smile. Enggak banyak. Third slide. 1, 2, 3, smile. Okay. And lastly, the last slide. Okay, last slide. 1, 2, 3, smile. Okay, thank you all. All right, our first session will be the lecture for the first session will be from UNISA, Ibu Wiwit Probowati. And before that, before I welcome our speaker for today, may I have your attention, dear students? For during the lecture session, please make sure that your voice is muted, but the okay. Okay, sorry, Armand, please mute your. Kenapa nggak bisa mute? Okay, so before we start our first lecture, I would welcome Eka Rismaningsi, our moderator too, to give a review of Bu Wiwit's CV. Time is yours to apa?
Baik, terima kasih. Izinkan saya untuk membacakan kurikulum vitae dari pemateri pada pagi hari ini, Ibu Wiwit Probowati, dengan nama Wiwit Probowati, S.Si, M.Biotech, dengan pengalaman kerja berposisi sebagai dosen senior di Universitas Aisyah Yogyakarta dari tahun 2016 sampai saat ini, dengan pengalaman pendidikan yaitu Master of Biotechnology di Universitas Gadjah Mada dari tahun 2011 hingga 2013, dan PhD kandidat Saga University di Jepang yaitu pada tahun 2020 hingga tahun 2022. Adapun beberapa jurnal publikasi 5 tahun terakhir yang dikerjakan oleh Ibu Wiwit Probowati, di antara lain yaitu ada dengan judul Narcissus Plants, A Melting Pot of Virus, Lalu ada pelatihan pembuatan nata de coco di perkebunan kelapa Desa Margumulyo, Kecamatan Siagan, Kabupaten Sleman. Lalu ada efektivitas pupuk cair Pseudomonas florens Agensia Pengendali Hayati terhadap penyakit mosaik tanaman kakao. Lalu ada pemberdayaan perempuan dalam produksi masker dan penerapan disiplin memakai masker bagi pekerja harian dalam penanganan wabah COVID-19. Terima kasih. Saya kembalikan kepada Ibu Sarfina Mutia Sanfa.
Okay, thank you, Eka, for the short CV. And before we begin, may I have just a few housekeeping notes for all of you. So if you have questions for the presenter or the speaker today, you may raise your hand or turn on your microphone after the lecture session. So I will ask you to raise your hands when the Q&A session begins, okay? So, and also during the lecture time, please keep yourself muted during the session. And if you have any technical questions or anything, please reach out directly to the chat room, as well as for the attendance for today will be given during the last session, in the end of the session. So you have to attend all of the sessions before you may take your attendance list. Okay, so without further ado, I would like to invite Ibu Wiwit Probowati to give the lecture for the first section. The time is yours, Bu Wiwit.
Okay, thank you. I will share the material.
Okay, hold on a bit. Maaf saya. Okay, we will. Oh, okay, I see, I see. Okay, okay. Can you share your screen now?
Okay.
All right.
Okay, let's start the lecture session. The first session is about in vitro plant tissue culture at home. I'm Wiwit Probowati. I will share about my experience about tissue culture, but at home. First, the objective of this session first is I will explain about the tissue culture defined, and then we will watching a video about the tissue culture at home, and after the end of the session, we can discuss something if any student can want to discuss something. Okay, tissue culture. Tissue culture is a collection or technique used to maintain or grow plant cell, tissue, or organ under the sterile condition on a nutrient culture medium of non-composition. Or we know tissue culture or in vitro culture as micropropagation. tissue culture is formed as asexual propagation. This is the fast method of propagation from a small sample like seed or organ or only leaf, small leaf tissue, or sometimes we can use a flower or root of the plant. In the tissue culture, we can produce a lot of plant can produce a new cultivar, can be multiplied rapidly with the good cultivar. Tissue culture is established at the establishment of aseptic culture. The condition is different than conventional culture, and the multiplication of plant tissue placed in the culture is explant. Taking the plant tissue multiplied in vitro and preparing for existence in traditional growing system. The basic of tissue culture is based on the theory of cell totipotent declared by Swan and Sladen in 1938. That one cell in the plant have can grow, become completely plant. This theory is differentiate with animal cell. In the plant cell, we can grow from seed and then from leaf or from shoot, apical shoot, and they can grow, become completely plant that have a shoot, stem, and then root. In the plant tissue culture, we need a medium to grow the plant or explant. The medium we can adjust by the objective of our research. For example, if we want to grow root, we can make a medium containing high auxin and low cytokinin, because auxin produced in the shoot apical and then transport into the root. And for objective research to first should grow, we can make the medium that containing low auxin and high cytokinin. And then if the medium of plant tissue culture have the same ratio of auxin and cytokinin, we can grow callus. Okay. After we place the explant into the medium, we can wait the growth of the explant, become shoot, and differentiate. And then in the end of the stage, the explant will grow root, become a completely plant. There are several micropropagation terminology. First, I always mention explant. What is explant? Explant is a plant part used for initiation. The plant part we can use is the important is the plant part is a meristematic part, like the shoot apical and then root or seed. And we also use flower or petal to as an explant for initiation, become embryogenesis. And then after the explant grow, we can subculture. Subculture is used for change the media because the explant growth very rapidly and massive. We can subculture the explant to subdivide the explant in the new medium. And then after the explant can grow, become shoot or root, we can transfer to move the culture into the new media. It means this is different become between subculture and transfer. To transfer medium is different medium. This slide is the step of tissue culture process phase. First is media making or sterilization, and then initiation, multiplication, rooting, and the last is acclimatization. First, in the laboratory, before I share about in vitro culture at home, this is the composition, the ingredient of medium that we usually use in the laboratory. This is the standard that the medium in plant tissue culture have a element macronutrient and micronutrient. Okay, and then we also can measure the pH and also the temperature of the medium, and adding the hormone, growth hormone like auxin, cytokinin, or gibberellin. In the laboratory, we need to scale, weight on the scale, every element of the medium, like N, P, K, Mg, S, and calcium, and also micronutrient. This medium, every element is expensive and complicated because every plant and every aim of the research have the specification of special need. So our innovation, we can culture of in vitro culture at home by using medium. We can change using agar. This is we usually this is food grade, but the composition of this agar is similar, like same. We can grow the plant or explant in this medium using medium agar. And for sterilization in the laboratory, we usually use autoclave. But in the laboratory, many experiment or tool is expensive. So at home, we can use pressure cooker or rice cooker to sterilize the tool or the medium. Okay, this is the media that we can use to at home to make a plant tissue culture media at home. The composition medium is sugar, tap water, or we can buy sterilized white water at the store laboratory store, and then fertilizer. We can all brand. This is up to you, whatever all-purpose soluble fertilizer. And then tablet inositol. Inositol is used as a growth hormone or to sterilize the medium. And then vitamin and agar flake. And the tool, we can use a pressure cooker for sterilize the medium or sterilize the tool. Stove, water, heat safe glassware, or jam bottle. While we waiting the media is simmering, we can prepare the jar. And then spray the work area with rubbing alcohol. Make sure they can clean and spray jar and lid with alcohol to sterilize and let the alcohol evaporate. First step is initiation. In the initiation step, we can take out the plant and then wash in the water and removal and disinfection of the explant by alcohol or ethanol. And then place the explant on the medium. The goal of this step is achieve a culture without contamination. So make sure that all the part of explant is sterile. Okay. This is the condition in the laboratory. We're preparing for initiation of culture. But at home, we can use workbench like a plastic or small room to work. And the next step is multiplication. Multiplication, the explant will produce vegetative shoot. The vegetative shoot can improve by add the hormone like cytokinin to promote shoot formation, and then we can use auxin to promote cell division and growth. And then rooting. Rooting is growing should produce advantage root. The hormone that we can add to promote the rooting is auxin. Okay. And then the last step, after all the plant is completely grow, shoot, and then root, the explant are removed from jar. Okay, this is the part of acclimatization. Acclimatization is moving the condition, like the media, from in vitro in the bottle or in the jar into the real media, soil, in the garden or in the field. First, explant are removed from jar and then plant in soil potting mix or perlite, and then start in high humidity and gradually reduce, because this step is very we have very carefully to touch very carefully, because there are different conditions between in the bottle or in the room with the condition in the field. Okay, this is the last session for define of in vitro culture. Reference of plant tissue culture at home. There are reference you can download, or if you cannot download, you can contact me. This is the book about home propagation technique at home. Okay. Let's practice in vitro culture at home. Let's watching the video. I will share the video.
Okay, dear students, while we wait for Wiwit to share her videos, maybe if any one of you have any questions, you may jot down or write it up on the chat box, okay?
Okay, while you prepare the question, let's see the video to make in vitro culture at home. Let me show you how to make the tissue culture at home. Before we start, make sure that all the condition of your workbench is sterile. First, I will mention several material. All material needed available and display in the desk experiment. First is agar, and then this is a fertilizer, and in the glass of sterilized, this is knife, scissors, scalpel, and plant. Hand sanitizer, and then alcohol spray. This is the fan to sterilize, bleaching, glass, and then white glove, and then this is a tissue paper, plastic container, and this is saran wrap or plastic wrap. And don't forget, we wear a facial mask. Continue. First step, we sterilize the workplace by disinfectant. This is very important because we need the sterile condition in our workplace. Disinfectant, we can spray using alcohol or ethanol in all the area. And then don't forget to turn on the fan. The fan is used for aeration, because in the laboratory we use laminar airflow that have air conditioner and aeration. So we can use the fan. And then clean, sterilize, and turn on the fan. Actually, in the laboratory, we use like the lamp or candle to combine between chemical sterilizer, that is alcohol, and then light or using candle. Okay. Don't forget to wear gloves and disinfect your hand and wear mask while working. And then next, boil the agar with filter water. Okay, and then stir it and wait until the media is boiling. Okay, wait. Then in the jar, we prepare a solubilized fertilizer. Here, approximately a half of the media. And then mix the agar with solubilized fertilizer until they are condensed and solid. And then stir to make sure that the medium agar and fertilizer completely mix. Stir and wait until the media solidifies. Okay, and then we prepare for the eggplant. First, we cut the plant into the small eggplant using scissor and knife into the small piece. We prepare for Clorox or bleach to sterilize the eggplant because the eggplant came from the garden or the field that have fungi or bacteria in the inside of the plant. So we sterilize by dip the plant into the Clorox approximately for 10 minutes. Then after 10 minutes, take out the eggplant from the Clorox. Take out the plant from the Clorox and remove the plant and rinse with sterilized distilled water 3 times. Okay, to make sure that the eggplant not containing a Clorox or bleaching. Okay. Place it in the paper towel or tissue paper. And let the plant dry. Okay, the medium is already solid. And then we can lay the plant or explant on agar. Where one are touching the media, make sure. After all the explant is one touching the media, we can cover the container with the saran wrap or plastic wrap. We cover it to minimize the contamination. And then the left is store the culture at the proper place to incubate at 16-hour photoperiod. Okay. Don't forget to observe for contamination and wait the plant to grow. To observe, we can see is there any bacteria or fungi growing in the media or in the plant. If there are contamination, we can change or repeat again the experiment. Okay, that's all the video. Thank you for watching. We have still have several minutes to discussion. Mrs. Mutia.
Okay, thank you, Ibu Wiwit, for a wonderful lecture. Okay, dear students, as you know, today is about the home culturing at home, right? Tissue culture at home. So if any one of you have any question, please raise your hand. You may directly ask Bu Wiwit. Normally in Indonesia, we call Bu as a replacement of like Mrs., something like that. So in Indonesia we call Bu. Kalau di Malaysia biasanya macam cikgu kan. Or kalau lecturer we use doctor ataupun professor, something like that. So okay, students, anyone who wants to ask questions? I think I don't see any faces of you here. Please open your camera so I could recognize some of you. Anyone wants to ask question? Okay, I see RD Lexon. Camera is on. Okay, any one of you, please, who wants to ask question?
Come on, ask something.
So any one of you is familiar with tissue culture? Maybe there's lots of biotechnologists here. Okay, any one of you students who wants to ask regarding tissue culture? You may ask Ibu Wiwit, yeah? You can directly raise hand and just unmute your speaker so you can talk and ask. Okay, before waiting for other respond, Wiwit, may I ask you one question? From your own experiment before this, perhaps if you've done this tissue culture, how many times, I mean, for the contamination, normally what kind of contamination it is if we try to use like the homemade tissue culture?
Contamination, talking about contamination, because we prepare the plant or the explant from garden or the field. Usually many fungi grow, especially contamination from the seed if we use seed.
Oh, okay. If you see, okay. So recommended part of the plant that can be used as an explant, which part is it the best for home tissue culture?
The important explant have embryonic or meristematic explant, like the shoot and then young leaf, okay, and the root. We use seed for easily, but very tricky to sterilize because the seed have a capsid to cover the seed, and this part is very difficult to sterilize. But if we can sterilize very completely, we can grow the seed very rapidly. So we can use seed very easily.
Okay, thank you for the answer, Wiwit. Okay, dear students, any of you want to ask any question? Since this is a type of lecture, so we need to have a discussion, okay? If not, I may not give you the the certificate. Okay, any one of you, please?
Or using bahasa Indonesia or bahasa Melayu, it's okay.
Yeah, sure. Boleh pakai bahasa Melayu, boleh pakai bahasa Indonesia juga bisa.
Oh, there are
one question from Shikili. How long it usually takes for the first growth?
For the first growth, usually only 3 days or 1 week to grow the seed or leaf. Around 1 week. If there are no contamination.
Does it answer your question, Shikili? Okay, another question coming from Effie Fever. Is it your name? Okay, dear madam, how to grow seedling resilient to diseases from the methods that is prone to infection?
How to grow seedling resilient to disease form, the method that is prone to infection. What the meaning? Can you explain?
So maybe in what I get here is like the seed which is resilient to disease. Is it different like from the normal seedings or how?
Oh, I see.
And
because,
especially the methods, is it different or not?
Oh, I got it. Because this is at home, in vitro culture at home, that the tool or the kit is different with the laboratory. So for this objective, maybe we need a culture in the laboratory, because in the laboratory the condition is very aseptic. Okay, and we can grow the plant with adding the hormone, additional hormone, to trigger the callus, because the seedling is resilient to the disease.
Okay, so does it answer your question, S.Si? Is that how I should pronounce your name? Okay, another question coming for Wiwit.
Okay.
Is tissue culture at home from Tivia Kartigayan? Okay, is tissue culture at home possible using a dying or an unhealthy plant from our background or garden?
If the condition of the plant is dying or unhealthy, it's maybe more difficult than healthy plant. But we can still use that explant, because the cell of the plant, that explant, that dying or unhealthy, still can grow and have the meristematic or embryonic plant. So it's okay. We can use a dying or unhealthy plant, because the strongness of the advantage of plant tissue culture or in vitro culture is to micropropagation very rapidly in the bottle, in the culture medium, that many plants are difficult to grow in the garden or in the backyard or in the field, or the plant is not difficult to grow, but rare, very rare, like orchid or several plant that is very rare and extinct. Okay.
Okay, is there any more questions? Because I think we actually have so many more time. We still have a lot of time. Or anyone here, maybe, dear students, who have tried to propagate or tried to do this tissue culture at home? Does anyone of you here have tried this experiment? May I know? You can share your experience here as well. Okay, maybe from Gigi, from the MP. A question here. Is there any effect to culture plant?
You mean the effect of the plant from by in vitro culture? I think no effect in the plant that using in vitro culture or conventional in the soil, because in the in vitro culture plant, we make condition that sterilize with the media that have a complete or good component of the media. So maybe the plant growth in the in vitro culture will be very good quality and produce the good cultivar, but the weakness of the plant growth in the in vitro culture is very risk to acclimatize in the soil, because we need to adaptation between the atmosphere in the bottle with the atmosphere or the real atmosphere or environment in the soil. You understand? Ciaci? Okay. The next question.
Okay, next question is, I think from Dr. Adila. So far, what kind of plants have you tried to culture at home, and what is the successful rate?
One that we try to culture at home is flower orchid that successfully grow and selling in the market, because in Indonesia now have orchid or monstera plant. But especially orchid.
Have you tried to grow yourself Wiwit at home? Or is it orchid as well?
For now, not yet. But in the around 2012, I have experience in the greenhouse of someone with growing orchid at home by plant tissue culture. For the variety or species or orchid, usually dendrobium. That's familiar. But for rare orchid, I never grow in the plant tissue culture, because dendrobium it's easy and many variant in the garden or in the forest. Dendrobium or Phalaenopsis, anggrek bulan in Indonesia.
That's very interesting. I think I want to try and grow dendrobium myself at home. Okay, anyone else want to ask question, or maybe actually could share some of your knowledge before this? Okay, another question, Ibu Wiwit dari Ziwei. What is the major difficulty in plant tissue culture method besides contamination?
Okay. Besides the contamination, the difficulty in plant tissue culture, especially at home, is to make the condition or make the media is appropriate or specified for the plant, because every plant have a different or special or specific need. For example, we grow orchid and in the bottle or in the plant tissue culture. We don't know the native composition of the soil in the forest that the orchid grow, and we adjust the media in the bottle by using agar. So that's the major difficulty. So we need to make many experiment like adjust the composition of the media, adding the how many hormone, how many composition of the agar, because different endemic plant, I mean the native composition of the soil in the forest or in the field is different with the composition of the medium. That's the major difficulty and very difficult. Sometimes the seed or the eggplant fail to grow because maybe the composition of the media very different with the soil in the native environment of the plant. Okay, this is question or
another question.
Another question.
So from Fever, what is the ideal environment temperature to culture a durian seed at home? I tried to grow it once at home, but died very fast.
Oh,
durian. Who
Are
likes
you-
durian here?
Are you trying to grow it in the soil or in the in vitro medium? Favor? Oh, in your mother Tupperware. Oh, okay. I will explain about my experience to growing the durian seed. I like durian seed or salak. You know salak? In Malaysia, is there any salak? Oh yes, a durian or salak or any other fruit that have difficult to grow or special environment to grow, like for example durian. Usually in the laboratory we will grow the seed of the durian by growing not the shoot apical meristem firstly, but we will grow the seed with the callus. Right. Callus is the culture plant that using medium the same or average between auxin and cytokinin. Callus is only cell that is undifferentiated of the cell. So for durian seed, we make a callus, trigger to callus, and then after the callus, we subculture and transfer the medium into the special medium that using auxin and cytokinin in the same volume. So we can grow the callus and differentiate the callus of the seed durian very rapidly, but only in the laboratory. If you grow in the Tupperware, it's impossible. Because for durian seed or you know jati tree have a special seed that the capsid or the skin of the seed is hard. So we need the special experiment like burning the seed of durian or the jati seed. So the skin or the capsid of the seed will be burning and open, and then the medium or the water can enter and absorb to the seed. Okay, Effie favor, understand?
Okay. Oke, ada lagi tanya tuh. Oh, in the lab only, madam? Or
Oh, yes, for making a callus that I explained before, callus from the seed, it's only in the lab. But if we want to grow in the soil, we need special experiment for the seed. For example, by burning or cut the seed. So the skin or the capsid of the seed can open and the water or the nutrient from the soil or the media can absorb or enter into the inside of the seed. Okay. But to grow trigger the callus of the seed, we only do in the laboratory. Okay, Effie.
okay, does that answer your question, Effie Fifa?
Try at home.
Okay, I think we have another question from Adam. Dear madam, in what form and how we can sell the homemade tissue culture product? Now, yeah.
In what form? Oh, the tissue culture product that we make product from home, homemade. The connotation is homemade is food. Tissue culture product from at home. We can sell the product. Maybe the best is using polybag. So the condition is acclimatized. After we make experiment in the jar or in the plastic jar, we completely grow the plant from tissue culture, and then after completely we gradually reduce the environment by transfer the plant from the agar media into the soil media. But usually the condition is we need to keep the humidity, especially humidity of the soil. And first, before we plant into the real area in the garden, we need the step by step by placing the polybag of soil, the media that we transfer from agar media into the soil, place the polybag in the room. In the room, and then after 1 week or several days, we can transfer the media into the real condition, like in the greenhouse or in the field. So we can sell the tissue culture product by the polybag in the soil media. But we need to give the label. This is different with other or conventional growth. This is product of plant tissue culture because different. Adam? Okay.
Anyone wants to ask again? Okay, from Marwah Amalia. Dear Madam, besides contamination, I've ever heard that browning in media is one of the challenge in plant tissue culture. Could you explain what it is and how to resolve browning in media? The first question, and then from Eka Rismaningsih, dear Madam, in a tissue culture course, I have tried to do this and there are several plants that grow and die even though they are in the same medium and under the same condition. So what causes it? Okay, maybe two questions first.
First question from Marwah Amalia. Beside the contamination, the media also can browning. Browning is caused by the evaporate or the evaporation of the explant that have several composition that can evaporate and browning. For example, polyphenol. So we need to transfer the new medium because in the media contain polyphenol that evaporate and then make the media become browning. Before we transfer into the new medium, we need to sterilize again the explant because maybe the explant not completely sterilized, like still have the polyphenol or cambium in the explant, so it makes the medium browning. Okay, answer the Marwah Amalia the question. Okay, and then the second is Eka.
Yes, from Eka.
I will read again. I have tried to do this and there are several plants that grow and die even though they are in the same medium and under the same condition. So the condition is already grow and then die in the same medium.
Yes, Madam.
I ever see this condition, but if we place several explant in one bottle, maybe it will make the condition of the medium like minimize nutrient for every explant. So for the solution after growth and then die, make sure that in one bottle we place the explant only one first, and this condition is caused by the nutrient is absorbed for many explant. So the nutrient will be sold out and then after grow and die. That's the answer. Okay. Are you interested to grow something, student? Oh, this is the.
I think we have another question from Chong Jiat Lee. Good morning, madam. May I ask which part of the eggplant is the best to culture a plant at home? Or it depends on the types of the plant. And before transfer to the plant to the soil, do we need to measure the pH of the soil first?
Okay. Several questions from Chongchat. Okay. The best part of the culture of plant at home. Nothing the best. Every part of the eggplant, of every part of the organ of plant have different characteristic. For example, if we use seed, we can grow very fast for the seed. But the challenge is many contamination. But if we sterilize very good, we can grow very fast of the seed. And then every part or every organ of the plant have different hormone inside. For example, in the shoot apical meristem have auxin. And if we use root or seed, we have, or if we use seed, we have internal hormone of gibberellin. And if we use root, we have auxin. So it's depend on the part of the eggplant. Every eggplant have characteristic because every eggplant have internal hormone or internal growth hormone. So have different characteristics. And then oh yes, and depend on the type of the plant. And every plant have different characteristics. Between orchid and maybe jati or durian is different. And before transfer to the plant, to the soil, better we need to measure the pH of the soil first, because if we move or transfer constant, like very rapid from the agar media and to the soil, that's make the environment change, so the plant will die. Okay, so it's be careful to transfer about the time and about the condition. Make sure that the condition, not only pH, but all, like the nutrition, fertilizer, same between the, if similar with the agar and the soil. Okay.
Okay. Next question from Nizar Abdurafi. Dear Madam, can it be applied for production scale and with the weakness of in vitro culture, such as the culture cost? Is it better than culture soil?
For question for Nizar, of course, in vitro culture, we can produce new cultivar that the food quality very rapid, and this very rapid, and the strongness is good quality of cultivar, and in the same time, very massive. We only need to prepare for sterilization and then the medium and the sterilized condition of the workplace. And then after all the plant grow, we need to prepare the soil media to transfer. The strengths of plant tissue culture than conventional medium or conventional plantation, we can grow very rapidly and massive at the same time of the plant. And then the second, the important, we can grow extinct or rare plant in the medium, which is in the native environment cannot grow very good. So we can save the world or save the plant. Save the biodiversity of the plant especially. Okay, Nizar, thank you. So the challenge in the plant tissue culture is only sterilized and medium and then the workplace, only that. After we solve all of them, we can grow and have the income from plant tissue culture. Okay, anyone? Any other question?
Any more question? It's okay to ask question as long as you can get more information from Wiwit. Okay. Even simple question can also, okay? All right. Any more question, perhaps, from the audience? Okay, so it means no more question, right, here from the audience? Everyone already understands about the tissue culture at home? Or maybe if you have any question regarding the slides. And also, Wiwit, may I ask you to send me the PPT slide so that I can share with the students later on?
Okay, after this session?
Yeah, sure.
Okay.
So, dear students, all of the presentations, the material, also the videos, we will share to you in a Google form, Google Drive later on, so you can actually download or and also see all the presentation again, as well as for our sessions. All the sessions are and will be recorded. So if you miss a little bit during break time, or maybe when you go to the product or anything, and some interruption with the internet, you may view again the presentation. Okay. And for the attendance, we will give it to you in the end of the session. So you have to stay until the end. So we will give you the attendance. So you can jot down your name so you can get your certificate. Okay. All right. Since Wiwit, maybe any more? Do you have anything to share to the students? or is it enough?
Okay, I think it's enough. Thank you for the attention and opportunity to share my experience about plant tissue culture at home. I hope this will be interesting to do at your home, because by doing in vitro plant tissue culture, we can save the plant from extinct for rare plant, and we can produce very rapidly and massive at the same time of the plant. Okay, and save the biodiversity. And for me, thank you, and I'm sorry if any mistake, and enjoy the international summer school. Thank you.
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Wiwit, for the wonderful lecture for the first session. We will start our next session, the second session, at 10:10 waktu Indonesia Barat, which is the Indonesian time, or in Malaysian time, it should be 11:10. Okay, so we will take a little break. So dear students, you may go to toilet or take some snacks or anything. And even during the session, you may have some snacks as well. You may drink and all, just like normal class, okay? So we will be back in 8 minutes. Okay? Thank you.
Tak perlu menjadi hebat untuk memulai. Untuk berani memulai apa yang kamu impikan. Keluar dari zona nyaman dan jelajahi potensi yang kamu miliki. Saat kamu terhubung dengan orang lain, itu menjadi kekuatan untukmu. Saat kamu memilih menjadi UNISA, kamu menjadi lebih dari yang sebelumnya. Menjadi kekuatan baru bagimu. Di seluruh profesi, konsistensi adalah produk dari etos kerja kami. Kami membuka wawasan, pengetahuan, dan pengalaman yang mempersiapkan kamu untuk bertarung di dunia nyata. Di sini kami merubah harapan menjadi peluang untuk mencapai sebuah tujuan. Selama lebih dari 3 dekade, kami hadir mewujudkan cita-citamu sesuai dengan perkembangan zaman. Konsistensi kami di dunia pendidikan memberikan cara baru bagimu dalam memandang kehidupan. Mewujudkan cita-cita bersama kami adalah cara terbaik kami memprediksi masa depan dengan menciptakannya untukmu. Kami meyakini bahwa kunci dari kesuksesan adalah profesionalisme, standar kualitas yang tinggi, serta mengimplementasikan nilai-nilai yang terkandung dalam Quran sebagai pedoman yang dapat mewujudkan tujuan pendidikan yang bernilai duniawi dan ukhrawi. Di UNISA.
Hai semua, hari ini
Okay, we are here for the next session, the second session of the day. May I ask Dr. Nazikussabah, are you here?
Yeah, hi, I'm here.
All
Okay.
right. Okay, before we start our next session, I would like to read Dr. Nazikussabah Zaharudin's CV. Okay, so for Dr. Nazikussabah, how can I call you, Doctor? In short.
You can just call me Naziku.
Naziku. Okay,
Yeah.
Dr. Naziku. So her research previously and also consultation on the food analysis, functional food, halal food, food safety, as well as analysis on bioactive compounds, human studies, as well as for her works currently related with seaweeds, enzymes, carbohydrate binding protein, food nutrition, type 2 diabetes, food labeling, food safety, and toxicity. Okay, so for her education, she had go through her degree from Bachelor of Applied Science in UITM, Mara. And also she directly go for PhD, isn't it, right? For her PhD in Food Nutrition and Wellness Studies in University of Copenhagen. And also a few of her publication recently regarding the effects of brown seaweeds, as well as seaweed-based palette binders. All right. So that is a brief introduction of Dr. Naziku. All right, without further ado, I would like to invite Dr. Naziku to give her lecture for today. Sorry.
Right.
I think co-host. All right.
Okay.
You are welcome,
Thank you.
Doctor.
Yeah. Thank you, moderator. Okay. I will share you the slide. Can you see my slide?
Yes, Doctor.
All right. Okay. Okay, thank you. So selamat pagi buat Mbak dan Ibu, Bapa serta para mahasiswa daripada UNISA dan juga UMP. Okay, saya Dr. Naziku. Okay, saya mengucapkan selamat datang. Welcome to Virtual International Summer School. Okay, so pada hari ini saya akan membentangkan a talk, give some talk about food biotechnology and halal food. Okay, so once again, okay, welcome. Okay, I hope you enjoy and have you gain some knowledge during this session. Okay, as the moderator mentioned before, okay, my name is Nazikussabah Zaharudin. So you can just call me Naziku. So I've been give a lecture almost 13 years from 2009 at the Faculty of Industrial Sciences and Technology, University Malaysia Pahang. So basically, this university is located near Kuantan, Pahang. Okay, so if some of you have come to Malaysia, especially Kuala Lumpur, so this university is located about 300 kilometers far away from Kuala Lumpur. So I hope later on, maybe next year, okay, we will have a face-to-face what we call summer school, okay, and then maybe we can come there in Jakarta, and the student from UNISA also can come to Malaysia to UMP. All right, okay. So for today's activities, okay, we will having two activities. The first one is a sharing session from me. So I will talk about food biotechnology, halal principle, okay, how this food biotechnology is related with halal food, and then some briefly information about halal market, globally halal market, issues in halal food production, and then what are the food biotechnology from microbial use as alternative sources for the halal food production. And then we will have a short discussion or Q&A with the students. So maybe I will give one question to the students. So we will discuss what we have here in Malaysia and what the students have in UNISA or in Indonesia. Okay. So this is the key terminology. Before we start, there are a few terms that maybe you need to know. The first one is halal. Halal means it's a permissible. Permissible means we can eat, okay, as a Muslim. Haram is a prohibited. And then there is a GMOs. GMOs is genetically modified organisms. Okay, I think most of the students from biotech program, they have heard about this term, GMOs, okay, GME, GMF, genetically modified food. And then we have rDNA or DNA, okay, it is a recombinant DNA, or it also known as a DNA sequence from one organism, and it introducing to another plants or animals or other organism. Okay, so this is the term that we might use later on. Okay. Okay, for the highlights for this lesson, okay, biotechnology can provide alternatives to many ingredients for the halal food industry. I think from previous years, from 1,000 years before, actually we have used biotechnology, or we applied biotechnology in order to produce or process food. Okay. However, currently, nowadays, most of the producer or companies, okay, they are using or apply this biotechnology in order to produce halal food. Okay, I will discuss or show you what is the application of biotechnology on the food. Okay, so you will get some ideas on it. Okay, and then secondly, all substances that are used in biotechnological processes need to meet the halal requirements for final halal product. Okay, so for halal product, in order for us to get halal certificate, all the ingredients, especially the material or the process used, okay, for example, here we use biotechnology process or materials, it need to come from the halal sources. Okay, so later on we will take a look what is a halal sources. Okay, and then the third one is some biotechnology companies have been adapting their production process according to suggestions from halal certifiers. So nowadays there are a lot of companies especially companies who produce food products, not only food, but pharmaceutical products, cosmetics, or personal care product. Okay, now they are trying to enter the halal market. So what they need to do is they need to meet the halal requirement, okay, to get the halal certificates. For example, we have in Japan, okay, they have a lot of food products, okay, where they want to sell or to enter the market in other East Asia countries like Malaysia and Indonesia. So they need halal certificate. Okay, so because they know that the consumers from this country, especially from Malaysia and Indonesia, are Muslim. Okay, so not only Japan, but other European country also. Okay, okay. So looking at the biotechnology, okay, I think most of you know what is the biotechnology. So it is a utilization of the organism or biological process for the benefit of mankind. So talking about food technology, so it means that we use organism, any living organism, and applied it in on food production. Okay, for example here we have yeast, okay, to make bread. Okay, it involves fermentation process. So this process we call as a conventional biotechnology. Okay, okay. These are the examples of conventional biotechnology, or we call as a traditional biotechnology. Okay, we have a bread making. So here the use of yeast to make bread, it involves fermentation process. Okay, and then we have a cheese making. Okay, here, okay, the cheese making is using rennet. Okay, rennet is a complex set of enzymes. Okay, and this rennet has traditionally been used to separate milk into solid and also liquid in order to produce cheese. So actually this rennet act as a coagulant, okay, to separate solid and liquid form. And then in the production of yogurt, okay, yogurt is a food that produced by bacteria, okay, through bacterial fermentation. So the bacteria we call as a lactobacillus, and sometimes we call it as a probiotic foods. Okay, probiotic foods is where it contains bacteria. Okay, and how it works, it is based on the fermentation process, where the fermentation of sugars in the milk by this bacteria will produce lactic acid, okay, which act on milk protein to give yogurt texture. Okay, so they act as a protein, and these probiotics actually are good for our health. Okay. And then the second example is the modern examples of food biotechnology. So today, biotechnology utilizes the knowledge of plant science and genetics. Okay, here we have genetic modification technology, and then we have also recombinant DNA or rDNA biotechnology. Okay, for examples here, okay, these two provide new technologies to target specific food, like for example, to increase crop yield, okay, to delay, for example, this tomato, okay, it is act as a this GMO act as an insect resistant for these tomatoes or any other vegetables or fruits. Okay. So these are some examples of food biotechnology. So we have two. The first one is traditional biotechnology or conventional, and the second one is a modern biotechnology. Okay. So any questions so far? All right. Okay. Yeah. Is there any question? All right. If not, I will continue on the benefits of food biotechnology. Sorry, this is a food biotechnology. So what are the benefits of the application of biotechnology on food? So many products in development are being engineered to confer nutritional benefits. Okay, for example, we have a golden rice, where this golden rice is actually called as a GMO, GMF, genetically modified food, where it contained beta carotene. And why it called as a golden rice? Because it is a yellowish color. Beta carotene is a yellowish color. Okay, and then this beta carotene produce vitamin A. So this vitamin A is one of the nutrients component. Okay, and it's good for health, especially in minimizing vision deficiency. Okay, nak kurangkan masalah penglihatan. Okay, so these are some benefit of biotechnology. Improve resistance, increase nutrients. Okay, and then not only that, it also enhance taste and also quality. For example, before we see that tomato, okay, so and the advantage of this biotechnology is it improve the quality, not only the quality and also the shelf life. So if we add some gene on these tomatoes or vegetables, okay, it's actually can delay or make a longer shelf life after harvest. Okay. So these are the benefits of food biotechnology. Okay, before we are going further, I would like to explain you what is a halal principle or what is meant by halal. Okay, halal is a food that are being free from these elements. Okay, what are the elements? Free from pork, free from dead carrion or blood, or all blood derivatives, or it contain low concentration of alcohol level, or any harmful materials to human health. So if it contain all this, so this we call as a haram. It is forbidden and identified as haram, not permissible in Islamic terminology. Okay, so for items with the halal certification, okay, usually kalau you pergi, for example, market, okay, you will see a lot of products. Some of the products contains halal logo. Okay, so food items with halal logo or halal certification have been processed by strict safety and hygienic guidelines. Okay, so why this halal is very important? Basically because this food we can guarantee that it is safe, it is more hygienic, and it also promote physical and also mental wellness. Okay, it is good for health. Okay, so this is the principle of halal requirement. It must be from all these materials or product. Okay. Okay, looking at the global halal market, okay, as you can see in this graph, from 2018 until 2028, okay, the global halal market increased in USD trillion, okay. Food that meets halal requirements have significant economical share of the global food market. So why? Because this is considering the increase in the population of Muslim communities. Okay. Not only that, okay, it also estimated that this size of the global halal market increase not only in food but also in pharmaceutical and cosmetic products. Okay, so this is the combination of food, pharmaceutical products, cosmetic or personal care products. Okay, so you can see that the demand are here. And if you going travel anywhere, for example, in Thailand, also in Indonesia or Malaysia and other country like Japan, European country, they also have some product that contain halal logo. Okay, so you can see that the demand are here, and not only Muslim can consume it, but other religions also can consume it. Okay. However, okay, in order to produce halal food, okay, we need to look at several factors, or it is known as issues in halal food production. So these are the important things that we must look at. Okay. The first one is a raw material. So we need to know the origin of the animal or plant and where does it come from, where we import that product. Okay. And then the second one is slaughtering. Okay. Slaughtering is a sembelih. Okay, sembelihan. Okay, so it must follow according to Sharia or using stunning method that follow the Islamic rules. And then the processing or equipment. Okay, of course, in order to produce a food product, it involves processing and also the utilization of equipment. Okay, not only that, we also use processing aid. Okay, processing it's like cleaning tools, and then like grease or oil. So we need to know what are the materials origin, okay, to avoid the contamination. Okay, and then number 4, food ingredients and additives. Okay, we need to know whether this food contained pig derives like pork, lard, or gelatin. Okay, and then we need to know the enzymes used, or where does it come from? Is it from rennet? Okay, and then the emulsifiers used. Okay, for example, if you see in the labeling or packaging, okay, you can see that if let's say it contains E471, it shows that it contains mono or diglycerides. So we need to know, okay, whether this product contain this mono or diglycerides or not. Okay, so we need to consider this one in order to give halal certificates or to produce halal food. Okay. And then number 5 is biotechnology and GMOs. This also is one of the issue in order to produce halal food, because we need to know what are the sources or the origin of the microbial, microbe use, or the organism use, okay, in the food production or food processing. Okay. Okay. And all right, talking about the modern biotechnology, genetic modification technology in food production. Okay, genetically modified foods are the foods that are processed by GMO. So however, the decisions on the GMO and the status of genetically modified food is still being discussed. We need, as I mentioned before, we need to consider what are the origin or the material or origin sources used, okay, in these GMOs. Okay, the discussion of the fermentation process that are done with GMOs and also the evaluations of recombinant bioproducts as food ingredient, it can be categorized under three headings. Here we can see that gene source, okay, we need to know where is a gene source. Gene transferred between living species. For example, is that transferred from bovine to plants, or is that transferred from animal to microorganisms? So we need to know the gene transfer. And then why we need to know? Because it has been reported that if a gene derived from non-halal donor, for example, like pigs, so this product, the final product, is not accepted as a halal product because we look at the donor. Okay, and lastly, the effects of the final product on human health. So we need to see whether this GMO is effects on human health or not. Is it safe? So they need to do toxicity tests, okay, to know or to identify whether these GMOs products are safe or not. Okay. Okay. Now, looking at this, okay, potential of microbiome for halal food production. Okay, so in food biotechnology, okay, there are a lot of potential microbiomes that has been applied. In order to produce food. Okay, especially during food processing. Okay, one of it is a protein based ingredient. Okay, and then the second one is red color substances. Okay, so considering that this microbiome production has many advantages, for example, fast growing of microorganism, and then it also produce in high productivity, okay, comparing to the other ingredient sources, okay, and also it is much lower in terms of cost, okay. So this potential microbiome is being applied for halal food production. Okay, so we will take a look one by one. Okay, what is the potential microbiome used in halal food production? Okay, starting from single cell protein, okay, until red color substance. Okay, so these are single cell protein. It is a protein base actually. Okay, as a solution for halal food production, biotechnology provides the following alternatives for protein-based ingredients. One of it is a single cell protein. Okay, What is a single cell proteins? It is considerable as alternative offered by this biotechnology, where from here the consumers can avoid the use of blood or porcine origin proteins. Okay. For single cell protein, the protein is derived from fungi. Okay, so this fungi is commonly called, protein is commonly called as a mycoproteins. Okay, so what are the application of this mycoprotein? It is basically usually used in human food and also to feed animals like fish. It is for aquatic or animal feed. Okay, so these microproteins are detected to be high quality protein, similar to meat protein. In USA and also European country, okay, they have given this legal approvals for some countries to sell foods with these microproteins produced. Okay, food enriched with microproteins also have been approved by a few halal certification bodies. Okay, what is a halal certification bodies? For example, in Malaysia, we have JAKIM, Jabatan Kemajuan Islam Malaysia, or Department of Islamic Development Malaysia. And in Indonesia, we have halal certification bodies called MUI, Majlis Ulama Indonesia, right? And then also in Singapore, in Thailand, in Japan, or in US, they have their own halal certification bodies. Okay, who will approve this food is halal or not by giving certificates. Okay. So as you can see in this picture, this is a tempeh. Tempeh is made from soya bean, okay, and also fermented with this fungi or filamentous fungi. So it also produce microprotein in it. Okay. Okay. Other protein-based produce is gelatin. Okay, this gelatin is actually derived from collagen. Okay, where we get this collagen? Okay, it's actually from animal connective tissue. Okay, it has various functions such as gelling and thickening agent. Okay, so the most used sources in food grade gelatin production usually come from porcine skins and bones, or from bovine skin and bones. Okay, as a promising gelatin type for the halal food industry, fish gelatin was manufactured. So to replace or to produce some alternative sources for gelatin, currently they are produced fish gelatin daripada ikan. Okay. However, this fish gelatin has a small share, or a few. That means the production is a few, small share. Okay, so how we want to overcome this? Okay, we will use a microbial for the alternatives to produce gelatin. Okay. So as you can see, this is some product used in gelatin. Okay, so we can use gelatin to make candy, jam, agar, or cake, or ice cream. Okay, so the most needed ingredients for the halal food industry is gelatin. Okay, so it's very difficult to find the halal gelatin. Okay, very limited. So hence of course, there is a lot of studies need to be done to produce this food grade gelatin, especially from the recombinant gelatin using, maybe can use bovine collagen, okay, in variety of microbial expression system. Okay. So to sum up, this recombinant gelatin produced using a halal gene source have become a potential source for halal gelatin. Okay, so these are the application of the biotechnology in order to produce halal gelatin. Okay. Okay. Another one, protein base, is a L-cysteine. Okay. L-cysteine is an amino acid. It acts as a building block of some proteins. Okay. It is one of the most common reducing agent in baking. So, siapa yang minat untuk buat baking, maybe you will use some product called as a L-cysteine. Okay. As well as enrich beef flavor. Okay. However, this L-cysteine, okay, some of it is derived from animal and even human sources, such as goose and duck feathers, okay, daripada bulu, duck or goose, or from human hair, daripada rambut, or swine bristles. Okay, so it derived from this. Okay, so we need to consider what are the sources of L-cysteine. Okay. With considering the halal requirements, it is forbidden for Muslims to consume the foods that are processed with additives derived from blister or any dead animals or human hair, and the feathers obtained from the animals that are not slaughtered with Islamic rules. So therefore, there are some research studies going on, okay, about L-cysteine production, where they are using fermentation process, and then they are using microbial, okay, in order to produce this L-cysteine, okay, the production of L-cysteine with bacteria, okay, and this have been certified by the other country. The use of bacteria to produce L-cysteine is certified by other country like in US, German, and also in China. Okay. And then last one for the protein base. Okay. It is enzymes from lipase enzyme, chymosin, and pepsin. Okay, these are one of the most widely used components or enzyme used in the food industry. So it can be obtained from animals, plants, and also microbial sources. Okay, plants and enzyme sources usually are generally regarded as a safe from the Islamic perspective because it comes from plants. However, if it is come from if this enzyme is come from the animal, so we need to check it. We need to make sure that it is not come from the prohibited animal. Okay. For example, there are a production of lipase, okay, that is used in lipid modification from porcine origin. So kita kena lihatlah what daripada mana lipase enzyme tu. Okay. And then sama juga dengan chymosin and pepsin. Okay. It is used as a coagulant in cheese. Ni macam renin tadi tu. Okay. Chymosin is renin. Okay. It can be obtained from animals that are prohibited or animals not slaughtered by the Islamic method. So we need to check this one. Okay, what are the ingredients or the sources of these ingredients? Okay. Because of this, Okay, the recombinant chymosin has been adopted as a good alternative as a halal to produce a halal food product. Okay, so there are recombinant chymosin. Okay, usually this recombinant chymosin is produced using genetically modified microorganism. For example, genes from calf or camel or goat. Okay. In Denmark, they have a Novozymes. This Novozyme is a major processor company that produce enzyme, microbial enzyme. So this is a big giant company in Denmark, Novozymes, that produce enzyme. Okay, they have agreed with IFANCA. IFANCA is a halal certification body in America. Okay, so what they did is they produce the halal enzyme to meet the demands of the Muslim consumers. Okay. And lastly, okay, we have food coloring. So these colorants are additives, like food additives, and it used in many areas in food industry. Okay, color substances which are classified, it can be classified as a synthetic or natural. For synthetic colorant, okay, it's usually stable than natural ones. Okay, but some of these synthetic colorants are banned by some European countries, US, Canada, and Japan. Why? Because it leads to carcinogenic and also mutagenic effects. Okay, for example, they have carmosin. Okay, carmosin is a red synthetic colorant. Okay, berwarna sintetik yang berwarna merah, carmosin. So this is prohibited in Sweden and Japan. Okay, because the issue, health issue. Okay, while that one is synthetic, while for natural color agent, okay, some of it are not exactly appropriate for human health and consumer preferences. For example, some of the food colorant, natural color, they are using blood. Okay, and then this actually considering this factor from natural and synthetic color. Okay, microbial color agents are introduced. Okay, actually it is obtained by fermentation, and it is an alternative to natural and synthetic based colorant. So basically, for food biotechnologies nowadays, they have produced microbial colorants. Okay, they using microorganisms like yeast, bacteria, and fungi, and also some algae species. Okay, and this actually has been reported as a new microbial colorants. Okay, because it can produce in a huge amount or large amount of production, big industrial scale. Okay, so these are the application of microbial in food protein base and also food coloring. Okay, as a conclusion, okay, so biotechnology and also microbial products are inevitable, cannot be avoided, okay, in the food industry. So usually nowadays food biotechnologies has been applied even 1,000 years before. Okay, dulu kita guna fermentation process in order to making bread or wine, but for today we have a lot of new technologies, okay, like using genes, for example, okay. And then biotechnological application have many advantages, and they can contribute to the halal food industry. As long as the processing aids or the food additive, food ingredients and methods that are used in the food processing are halal. Okay. For a microbial production, okay, a microbial production is a precious option for the halal food producer. Okay, now this is very important, okay, in order to supply component such as gelatin. As we discussed before, we have a lot of protein based like chymosin, okay, gelatin, okay. So Muslim consumers are concerned about this component because some of it are derived from non-halal sources. So if this genetic modification process meets halal requirement, then it can be possible to produce various halal food product at a large scale or high efficiency. Okay. And last one, comprehensive research, okay, related to halal food biotechnology products should be done commercialized widely. Okay, I think most of you as a student later on, you will be the one who will continue to do this research. So if some of you are excited to do this kind of food research, halal food research, then it is a good, because nowadays it is a demand from other country to get a halal ingredients, and it also can be commercialized widely in other country or globally. Okay. All right. I think that's all from me. Okay. Before I answer some questions from you, I would like to ask all of you, can you give one example of food products developed through food biotechnology? Okay, you can give some example whether from your country or any other countries. So give one example of food product that are using food biotechnology.
Okay, dear students, you may jot down in the chat box what kind of food of biotechnology. All right, there's cheese. Oh, tapai. Soy sauce. What else?
Let me see.
Ketchup. Yakult. Tempeh. All right.
Good. All right. So this shows that you are not sleepy. You are still awake.
Kimchi.
Good.
Oh, okay. Korean
Okay.
food. Else? Nata de coco. Okay.
So cheese, yeah, tapai. In Indonesia, do you have tapai?
Yes.
Do you know what is tapai? Is it the same word used
Yeah,
in
the same
Indonesia?
word.
Tapai. Okay, from rice, right?
Yeah.
Fermentation process. Okay. Yeah, we have soy sauce or ketchup. Okay, good. That one is a fermentation process. Yes, we have Yakult. This is a probiotic drink where they add bacteria inside this beverage. Ika, tempe. I think in Indonesia you have a lot of food that contain tempe. In Malaysia also we love to eat tempe, especially with ayam penyet.
Yeah.
So we have here in Malaysia, we have a restaurant ayam penyet. So usually they will serve this ayam penyet with tempe. It's a good meals actually. Okay. Say good. Okay, there are lots of students give an examples on tempeh, yeah, kimchi. Okay, I think yeah, some of you like to eat kimchi. I haven't tried it, but maybe later on. I do not know what is the taste. So is it
Okay.
sour? Okay.
Oh, doctor, you need to try kimchi. I think the UMP students can bring you to eat Korean food.
Oh, okay, yeah. Okay, good idea. Okay, and then we have nata de coco. Well, this is using gelling agent, I think. Nata de coco using gelling agent. Blue cheese. So I think this is the first time I heard about blue cheese. Maybe that Shini can give some examples or explanation on blue cheese. Is it same with the cheese that supply in the market, or is it different?
Hello, Dr. Dashni here.
Yeah.
Regarding the blue cheese, recently I saw a YouTube video. So in that video, they originally make this cheese and then they inject that cheese with enzyme, I think. So I couldn't remember what enzyme, the enzyme's name. And they let it ferment. So and then after a few months, I guess, surround it, we can see like a lot of molds in blue color. We can see this animation thing in Mr. Bean's one of the series. So that's
I
when I got
see.
that idea. Oh, so they have this blue thing, blue cheese thing in reality.
So blue cheese shows that this cheese contain mold after a long time. Is it?
Yeah, it produces this color pigmentation.
Color pigmentation. Okay, nice. Okay, I would like to see this video. I will search it in YouTube. Okay, thank you for the info. And then we have from Weni Arun is Tauchu. What is Tauchu? Any one of you, Weni, can explain? Weni, are you here? Weni Arun, Tauchu or Tauco? Atau mungkin saya tak familiar dengan this food. Okay, okay. Tak apa, nanti saya akan Google what is a tauco. Okay. And then yes, making wine. Okay. Alcohol beverages from grape. Okay, fermentation of using a bacteria from sugar to alcohol. Wine, yogurt. Okay, natto, tansuki. Can you explain what is natto? Suki, are you here? Natto. What is natto? Any one of you? No? No, this is the first time I heard about natto. Okay. Tadi tauco, now it's natto. Okay. I will search for it. Okay. And then we have keju, yeah, cheese, kombucha, yeah, kombucha tea. Okay. So siapa yang minat minum kombucha tea, it is also involve fermentation process. So basically, it is a conventional biotechnology. And then the making of vinegar, okay, cuka, mentega, butter. Oh, natto is fermented soya beans eaten in Japan. I see, okay. So it's like a ketchup, but maybe is it in solid form or liquid form, this natto? So like ketchup is also, oh, okay, in solid form. All right, okay. So I think this young fella knows a lot of food from other country compared to me, the older one. I only know the traditional food. Okay. All right. I think that's all. Any question from the rest? Okay. Also fermented by beans used in Chinese dishes. I see. Okay. All right. Okay. Thank you for the info. Okay, if you have any questions, okay, you may just turn on your mic or chat it, type it in the chat box. Okay.
Any questions, dear students? You may directly ask Dr. Naziku, or you can jot it down on the chat box, in the chat box.
Is it okay if I show you a video about food biotech?
Sure.
We still have a time, right? Okay.
Yeah.
Okay, let me share with you. Okay. Oh, okay. Right, can you see the video? It's a white color.
Yes.
Boleh nampak?
Boleh.
We'll play it.
You need to open the view option and then you share the sound.
Sorry, how is it? Macam mana?
There's this view option from the sharing, the lower section of the sharing. There's like this label, and then you click that one. Wait.
Okay. Is it okay? Share screen, and then we have.
You share your screen first.
Okay. And then we have. So I just click screen, right?
Yeah, share screen first.
Okay, can you see it?
Okay, try.
Try. Is it working?
I think the voice is not shared yet. So maybe you can go from the share screen, the middle label. Then you can choose advanced sharing option.
Okay. Video, is it?
Share your screen, and then you can, after you share your screen, you can pick the label, the notification.
Is it advanced?
And then you share your screen first, and then you can click on the notification label, something like that, that goes out, like there's your mute, your stop video, and everything there.
Okay, let me try.
But you need to share your screen first.
Oh, I see. Okay, so this is my screen. Okay.
Okay.
I have shared. Okay.
And then on
can
your
see.
screen, you actually can see this like view option. Or di luar dari ini, dokter. Kan ada
Advanced
macam
sharing.
Advanced sharing.
And then multiple participant.
No. Soalnya enggak.
Start share. Oh, I think it's okay. I think I will share the link with the students. Okay, later on about the food.
It's okay. Or let me share it for you. What's the title of it?
Right. Okay, good. I did.
What is it? The title on the YouTube?
Okay, I share you the link.
Okay. Okay, I will share.
Makan 57% noda hitam. L'Oreal Paris Glycolic Bright. We're worth it.
Okay, can all of you listen to the sound? Now?
Yes.
Okay.
Yeah. Yeah, so basically that's just basic information or the a summary of the food biotech. So as you can see that it involves a lot of genes, okay, and some of the sentences they have mentioned about the crop yields. So this food biotechnology not only for the final food production, but also from the first they start to harvest from the crop, okay, and also application on the food processing until the final food production. Okay, so from start, maksudnya dari A to Z tu food biotechnology ni kita telah gunakan lah. Okay, daripada kita tanam tanaman from crop, okay, and then kita bawa ke kilang for the food processing, and also last one for the final product. For example, you have gelatin, how you use it at home, this kind of final product. So all this involve food biotechnology, and some and all of this if it is related to halal food, so it must be halal. That's mean from A until Z, from the crops until the final product. Okay, all right. I think that's all. Yeah, any question? Okay.
Any questions from the floor? Students, anyone wants to ask? The session haven't finished yet, so you may ask some questions now. Okay, before the students ask, may I ask a question, Doctor?
Yeah, sure. Okay.
So in your opinion on or based on your research previously, how much is actually, I mean, the percentage for the halal in our food it is, the current food chain? Is it like in Malaysia, for example, in Malaysia, is it like the food presented in the shopping malls and everything, is it already basically 100% certified halal or not?
Okay. Okay. Usually food that are available in the market, they have the one with the halal logo, and the one is without halal logo. So with halal logo, if the product is with the halal logo, that means this product is halal certified. So how we want to know this product is halal certified? The first one is based on the logo, whether it is from, for example, from Jaki Malaysia, or some of it maybe from, for example, the product from Indonesia. For example, the logo is from Majlis Ulama Indonesia. So in if it contains this logo, then it is 100% halal, because the panel and the auditor actually, they have gone through and checked all this information about halal and non-halal sources for the food product. They will go to the factory, for example, and then we'll see the process and collect all the documents to make sure that all the things are halal, and then they will give the halal certificate. However, there are food in the market sold that do not contain or have halal logo. It doesn't mean that the food that without halal logo is not halal, but sometimes some of the food, for example, they required some time to apply for halal certificate. Okay, untuk kita apply sijil halal pun perlukan masa So ada makanan ataupun barang yang dijual di market tu dia ada logo halal, tapi tak semestinya dia tidak halal lah. So basically in Malaysia, most of the food boleh kita katakan 80% saya rasa lah contain halal logo. Even it comes from a different company or factory. They, for example, for example, in Malaysia, we have a different types of food and beverages, okay, imported from other country. Usually kita akan lihatlah ataupun kita akan divide this food, katakanlah dia halal, we will put at a certain area. If it is a non-halal product, usually kita akan asingkan, put in a different area. So basically the product enter or market in Malaysia, I think 80%, more than 80% contain halal logo, halal certified. Okay.
All right, thank you for the answer, Doctor. Maybe any other question from the floor, students? Ke dah mengantuk? Anyone wants to ask?
Maybe I share some experience with you. Since you are students from Biotech program, I think it's a good one for you because you have a knowledge. Later on, bila you all kerja to find a job, you can go not only doing a research lab, but you can also be a panel, for example, for this kind of things. For example, in Japan, they have a Majlis Fatwa panel to recognize or give a halal certificate to the companies like food or cosmetic or pharmaceutical companies in Japan. So what they required is they want a panel or auditor from a technical person. This technical person need to have a science background, especially from biotech or chemistry background. So when you act as a panel, so you should know what are the process involved, fermentation involved, the enzyme used, the microbe used. So you have some knowledge on it. So that are some things that you can consider later on after you finish your work. Okay. Some application of your study in a nanti bila you kerja lah in a real job, okay, your future.
Okay, Doctor, I want to ask one thing. Is that, like, for example, when we use fermentation, when we do fermentation using bacteria, how actually, like, the bacteria itself is considered as halal certified?
Yeah, okay. Actually, in order we know that this bacteria is halal certified or not, we need to know the source of this bacteria. For example, if this company produce or if this laboratory produce this bacteria, they need to show us the certificate of analysis. Where does it come from? What is the origin? And if we have this certificate of analysis, then we can give we can consider it as a halal bacteria, halal sources. So actually, it must come from the lab or the company itself. So they need to give us some documents to show or to give some evidence to us. Okay. And in the other hand, let's say, for example, kita kan walaupun ada dokumen yang mengatakan this bacteria is from these sources or these non-halal sources, we also can actually, for example, do some analysis. I'm not sure how, but maybe some of you, I think doctors or professors knows how to detect this bacteria, what is the origin or the sources. Okay. Okay.
Okay, we have some question here from Marwah Amalia as well as Suci Indah. Okay, so from Marwah Amalia, I'm so fascinated by your insightful presentation, madam, especially in microbial potential for halal food production. As Muslim, of course, we should consider any food properties as safe and halal. Could you explain what is the critical point to make micro products categorized into halal?
Okay. All right. Nice. Good question. What are the critical points to make micro products categorized into halal? As I mentioned just now, with this MC or moderator, okay, the critical point is, the first thing is we need to know the origin or the raw material used in order to produce this halal product. In order to produce the product, we need to know the ingredients. Of course, if we buy some food, we see the labeling on the packaging. It doesn't mention about the origin of enzyme. They didn't mention about the fermentation process. They just mention about the basic ingredients, what is in this ingredient in that food. But to get a halal logo or certified halal, the critical point that the auditor or panel looks are the bacteria origin. As I mentioned with the moderator before, we need to have a look at the certificate of analysis from the lab or from the company. So the critical point, we need to look at the origin material or bacteria. Okay. Okay. And then I think there is another question from Suci Indah.
Yes,
okay,
from Suci Indah.
So what is the negative impact of overuse of biotechnology on the food industry? Yeah, of course, there is advantages and disadvantages on using these technologies, not only biotechnologies, not only for bio, for chemistry. If you use a lot of chemicals, even though it is nutritionist and give a lot of nutrients, but if we exceed the limit of using the nutrients, then it will give some toxicity. It will create some toxicity. For example, in chemistry, if we eat a lot of vitamin A or vitamin C, exceed the limit, daily limit, then dia akan mempengaruhi kita punya health lah, kesihatan. So same as a drug, pharmaceutical drugs, okay, there are a daily usage or daily limit yang kita boleh guna makan. Okay, so sama juga dengan biotechnology. Of course, there is a negative impact if we use it. For example, kalau you baca about GMO, genetically modified organism, okay, they have some advantages and disadvantages on using it. Some of it will create a health problem, okay. So tu yang kita kena lihat lah, kena kaji. So siapa yang kena kaji? We as a researcher in the university and also students from the university. So when you do a final year project, so maybe you can do some of this project to know, okay, whether this, for example, the advanced biotechnology is good or not for health or for safety, for example. Okay, so maksudnya akan ada continuous research lah on that. Of course, kalau saya sebagai researcher, katakan saya introduce, okay, saya introduce this gelatin is good, bagus, dia dihasilkan daripada this bacteria or microbe, for example, melalui proses this one, for example lah. But then saya ada continuous study, saya kena buat study juga, apa effect this kind of bacteria into human health. Maksudnya ada human study, ada animal study. In European country, for example, or in US, dia ada satu badan di mana bila you all nak guna atau nak apply this GMO, for example, katakan you nak guna gene or from this bacteria, for example, you kena buat several study. For example, tadi previous lecturer ada sentuh pasal in vitro study. Okay, so dari situ you boleh tahulah what is the negative impact of this biotechnology on food or other agriculture. Okay, itu contoh, dia mesti ada negative impact dia, tapi kita kena buat study dan dia tak boleh lebih lah penggunaan dia. Okay. Okay, can we consider all plant-based product as halal food? Yes, boleh. Sama juga dengan all plants you can consider as a halal food. Thank you, Kili, okay, for the question. But we need to consider the process involved. Is it involve the fermentation process? For example, in the making of wine, kita nak buat wine kan, alcohol beverages. It is come from grape, daripada anggur, buah, okay. But then why it is become non-halal product? Because it involve the fermentation process, where it involve certain bacteria, and the alcohol level is exceed the limit. Okay, itu contoh lah. But for your question, plant-based product as halal food, yes. But we need to look at the process involved, whether it involve fermentation or not. Okay. All right. Any question so far? It's a good thing. Okay, when all of you ask some question. Okay.
Any more questions, dear students? It's getting interesting. You keep asking questions, it's going to be more interesting. Okay, another question from Eka Rismaningsi. Dear doctor, what do you think about GMO food, which is currently traded in the market? For example, like soybeans as well, and then what else? Corn.
Yeah,
something like that.
Yeah, it's a lot GMO product in country. But as I mentioned before, yeah, of course there is some advantages and disadvantages and some limitation on using this GMO. But to produce GMO food, of course there is bodies like what we call FDA or FAO, food, something like that. It is a bodies that recognize whether this food is suitable or not to be consumed to other consumer. So kalau kita baca a lot of articles, some of it mention that these GMO products cause, for example, this for example, I don't know, cancer, something like this. But to claim about that, they need to do a human study on it. Kena ada buat human study lah. and then kita boleh claim lah whether this GMO food is good, safe to consume or not. If not, dia akan ada satu badan daripada, for example, FDA lah, dia akan I'm not sure FDA I think FDA, okay, food safety like that, okay, dia akan inform lah, okay, this food is not safe to be consumed, and dia akan ban that GMO food, okay. Alright. Okay. Yes. When we talk about food ni sebenarnya, it's not. It's about the raw material. Okay. The raw material we use and what are the ingredients we use during the process, the final product, and also the safety or the effect of this food product on human health. We need to study. Okay, so kalau but for food is not required. I mean, it is not compulsory for them to do human study. But if you want to produce, for example, drugs or pharmaceutical products, then you need to do human study to confirm or to claim this pharmaceutical product is safe or not. Okay. All right. Okay. Thank you, Ika. Okay, for the question. Anyone? Any question? Okay. So in Indonesia, lambat 1 jam ya?
Yeah, Dr. lambat 1 jam.
Alright, I see.
But here already Zuhur time. Just
Zuhur
now.
time, okay. I see. So.
Okay, so any further questions from the floor before we end our session for today? Okay, let me remind all of you that in the chat box, there's this attendance form that you may fill in, okay? So who haven't filled in the attendance form, please fill in now. Okay. Okay, no more questions from the floor? Anyone? Okay, since there's no more questions for this session, thank you, dear students, and thank you to all the speakers for today. This is our first day, our first and second session. So we will have the second day, third day, and the fourth day for this international summer school. So I hope all of you will attend the course because we will give the certificate based on each day courses. So when you attend 4 days, it means that your certificate will show that you attend 4 days of the event, something like that. Okay, so if only 1 day, 2 day, 3 days, so it depends on all of the attendance. Okay, so we will record all of the attendance for each day. So hope all of you can attend fully so you can get a full certificate. All right. Okay, I think thank you so much also to Dr. Naziku for this exciting about halal food biotechnology. And thank you, dear students, for attending the first day. We will see you again on the next day with the same Zoom link. So we will see you tomorrow at 8:30 Indonesian time. So it should be 9:30 Malaysian time. So, okay, that's all for today. Thank you very much, dear students. See you again soon. Bye. Assalamualaikum warahmatullahi wabarakatuh. Thank you so much,
Thank
Doctor.
you.
Thank you so
Yeah,
much.
thank you. All the best to all of you. Okay, thank you.
Okay.